I'm Ken Wang from Cleveland Clinic. I'm a geneticist working on atrial fibrillation. Today I'm going to talk about an unexpected finding that links nuclear pore malformation to atrial fibrillation. I'm going to cover three major topics: genetics of an interesting AF family, nuclear pore complex, and the molecular mechanism by which loop 155 mutations cause AF. Back 12 years ago, we studied a very interesting family from South America. The family had 15 family members and 5 living generations. The age of family members ranged from 3 months to 93 years. Interestingly, there were 7 consanguineous marriages in the family, and there were 6 patients with AF. They all were derived from consanguineous marriages, and their parents were normal. So this is a typical autosomal recessive AF family. The proband had chronic AF and QTc of 0.42 seconds. He was delivered by C-section due to fetal tachycardia and atrial fibrillation. Echo at day 2 showed marked dilatation of both atria and ejection fraction of 52%. EP at 1 month detected AF. Echo at 2 months, however, showed mild atrial dilatation and a normal ejection fraction of 52%. The patient died suddenly at the age of 15 months. Another interesting patient in the family. This patient had chronic AF and a QTc of 0.4 seconds. The heart rate was normal with medication. Echo at 15 months was completely normal. However, the patient died suddenly at the age of 19 months. There are several other interesting patients in the family. And so for the next 4 years, we focused on this family and worked very hard to find the gene that causes AF in this family. And our hard work was paid off. And finally, we found a mutation in this gene called loop 155. The mutation is a arginine to histidine change. And the gene is, as I said, is loop 155. It encodes a nuclear porin protein. Nuclear porins are very important proteins in the nuclear pore complex. Here's the putative structure of the nuclear pore complex. And then loop 155 is localized in the central part of the nuclear pore complex. Loop 155 is on chromosome 5p13. It encodes a protein with 1,391 amino acids, and its molecular weight is 155 kilodalton. That's why it's called loop 155. Next, I'm going to give a brief introduction on nuclear pore complex. During evolution, the hallmark of eukaryotic cells is the development of the nucleus and compartmentalization of the genetic material inside the nucleus. There are many pores on the nuclear membrane. In each cell, there are 3,000 to 5,000 nuclear pore complexes. And each nuclear pore complex consists of 50 to 100 proteins. The major function of the nuclear pore complex is nucleocytoplasmic transport of macromolecules. For molecules with a molecular weight of less than 40 kilodalton, they can go through the nuclear pore freely by passive diffusion. However, for molecules with a molecular weight of more than 40 kilodalton, then energy-dependent transport is required. As we know, mRNAs are made in the nucleus and processed then they have to be transported into the cytoplasm to execute their functions. For example, mRNAs need to be translated into proteins. Then some proteins, for example, transcriptional factors, splicing factors, they have to be transported back inside the nucleus to execute their functions. Therefore, the major function of the nuclear pore complexes is to regulate exchange of molecules between the nucleus and the cytoplasm. The selective traffic of proteins and RNA through the nuclear pore complexes plays a critical role in regulating eukaryotic gene expression. Now I'm going to talk about the molecular mechanism by which loop155 mutations cause AF. Loop155 is highly expressed in the heart. We overexpressed the mutation we identified, R391H, into a cell line, in this case HEK293 cells. We tagged wild-type or mutant loop155 with either EGFP or FLAG, and then looked at their cellular localization. In the wild-type case, we can see clearly that loop155 is localized on the nuclear membrane, and it has this kind of dot-like staining. This may be the nuclear pore complexes. However, the mutant loop155 is mostly localized in the cytoplasm, and the distribution is most smooth. Then we also characterized the loop155 mutation using a newly developed method for nuclear permeability. Compared to the wild-type loop155, the mutation significantly inhibits the mutation reduces the nuclear permeability. Then we created knockout mice for loop155 to further explore the molecular mechanisms for atrial fibrillation. Unfortunately, homozygous mice died during embryogenesis. However, heterozygous knockout mice are viable, so we carried out studies in heterozygous mice. This slide just shows that expression for loop155 in heterozygous knockout mice is reduced by 50%. So we recorded EKG from wild-type or knockout mice. And in the wild-type case, we can see very clear and regular RR intervals. And we can see clear P waves. However, in mutant mice, we detected this irregular RR intervals, and in all these traces, P waves are missing. So these are classical characteristics of AF. We further characterize this knockout mice using intracardiac EKG recording. In the wild-type case, we basically placed a catheter with electrodes in the right atria and right ventricle. In the wild-type case, with stimulation, we didn't detect any abnormal electrical activity in both right atria or right ventricle. However, in the knockout mice, with stimulation, we can see clear abnormal AF-like electrical activity. And in the right ventricle, there is an irregular R interval. Again, these studies demonstrate that the loop155 knockout mice develop AF. Next, we used some in vitro studies and knockout mice to test whether mutant loop155 affects export of a heat shock 70 mRNA or nuclear import of a heat shock 70 protein. Heat shock 70 is just a marker in this case. First, we did studies in Hela cells. We used RT-PCR analysis to mirror the ratio of cytoplasmic RNA versus nuclear RNA. This ratio is an indicator for nuclear export of messenger RNAs. As shown here, compared to the wild type loop 155, the mutant loop 155 had reduced nuclear export of the heteroscopy mRNA. Then we used the immunostaining to check the protein import. And as shown here, compared to the wild type, the mutant loop 155 significantly reduced nuclear import of the protein. Identical results were obtained with neonatal myocytes isolated from wild type or loop 155 knockout mice. And again here, RT-PCR analysis showed that reduction of loop 155 by 50% significantly reduces nuclear export of heteroscopy mRNA. And the knockout also reduced nuclear import of heteroscopy into the nucleus. Next, we try to find a cellular mechanism for the AF in loop 155 knockout mice. So in this case, we isolated cardiomyocytes from mice and recorded action potential durations. Compared to the wild type of mice, loop 155 heterozygous knockout mice had significantly shortened APD lining. So what is the mechanism for APD shortening in loop 155 heterozygous knockout mice? We know the action potential is formed by many different ion currents. So theoretically, we think that increased IK1, IKI, IKs, and ITO potassium currents should be able to shorten the action potential duration. And also reduction of the sodium current and calcium current could also shorten the action potential duration. So this is going to be the focus of our future research. We are going to record all these electrical currents from wild type and knockout cardiomyocytes and find out which current is affected. So in conclusion, we have demonstrated that nuclear pore complex dysfunction causes atrial fibrillation in both humans and mice. AF causing loop 155 mutations reduce nuclear permeability, nuclear export of mRNAs, and nuclear import of proteins. Loop 155 deficiency shortens action potential duration, which may promote re-entry and atrial fibrillation. So we can conclude that the nuclear pore complex plays a critical role in cardiac physiology. And this is the first report to link the nuclear pore complex to cardiovascular disease and to cardiac physiology. And most importantly, we think loop 155 is a new therapeutic target for AF upstream of ion channels. In the past, people have focused on ion channels to develop therapies for AF, but with limited success. So I think focusing on an upstream target like loop 155 may offer very nice alternative to develop therapeutic agents for atrial fibrillation. These are the people who contributed to this work, which was published in Cell, and Yuhua Zhang and Yiqing and Tate did the intracardiac electrophysiology studies. Thank you very much.